work by many people that I'll try to keep the names up as their contributions come up. The method that I'm very excited to tell you about is still a work in progress, and I'm hoping it's widely applicable. The system that we're applying it to right now is T-cell, but we're hoping that this extends beyond T-cell. So this is my cartoon of the T-cell. It's like many cells in a context in situ, it can be thought of as an input-output machine. So that means that there's some input. In this case, it's in the form of a peptide antigen that enters the T cell via a molecule on its surface called the receptor. There's the T cell receptor, and then there's many other species of receptors as well. And then something happens, something else happens, something else happens, and then there's the outcome or the output. And the output can be the T cell going to kill another cell. That's part of its job as an immune cell. It can proliferate, so it can make more T cells. It can release cytokines that communicate with other cells, or it can just put molecules on its surface. And this includes molecules like CD69. There are ongoing efforts to engineer T cells. And so what I'm showing in purple is a human engineered receptor called CAR, chimeric antigen receptor. And these are now approved and in usage in human patients. The idea is that they want to improve the function of T cells in order to help them do their job in the immune system. And in part because of this, but also just because we want to understand T cells, there is a huge amount of potential for quantitative predictive models of T cells that go all the way from input to output. So given an input, can you predict quantitatively what the output is? And because this is an important problem, many groups have worked on it, and so many more than just what I'm showing here, but two of my favorites are one by Melissa Lever and Omer Dushek's group, and then work by the Finley group in USC. Right. So I will tell you about our effort to try to make an input to output model. This has not been what we have been working on traditionally. What we've spent sort of the last several years doing in my research group is focusing on an individual step. And it turns out that if you pick any step, then there's something interesting in terms of the physics of what's happening. If you sort of zoom into any one of these green dots, and that's because there are effects like, for example, hydrodynamic interactions between the two cells. This is something that John and I worked on. There are mechanical properties of the membranes that resist bending, et cetera. And then a lot of our efforts recently have been on the fact that many of the molecules involved are disordered. They look like big pieces of spaghetti, and that means they have high entropy. And of all of this work over these many years, I think one way of summarizing it all is that these are all cases where the system disobeys the law of mass action. So what is mass action? It's that if you want to find out how fast something is happening or how much something is happening, then you take the constituent ingredients like A and B, you find out what the concentrations are, and you multiply the concentrations, and you put a coefficient or a prefactor in front. And to really not much surprise, there are so much more interesting things happening, and that's in large part because of mechanics and because of entropic effects and many other things that it turns out to be more nonlinear than this. Okay, so that's what we have been doing, and in a way, that's the opposite of what you might call systems biology, where we want to integrate across this entire cell. Okay, so we started doing this, and one of the reasons is that there's now many experiments in a range of conditions, and our goal is to try to get models that match over a range of conditions and are truly predictive. So I will now spend 2 slides telling you about one data set. This is a published data set. The researchers, multiple researchers, thought of doing this at the same time. They said, let's try to understand this input-output system, not by changing the input, but rather by changing the time series of the input. So what they do is they introduce the input at a certain time, and they measure an output, which I'm labeling C here. They take the same input and just turn it on and off, on and off, on and off, measure the output, and they change the period. And in fact, there's 2 periods they can change. They can change the overall period, or just like the on period or the off period. And there's sort of 2 independent degrees of freedom there. And then measure the output. So the output could be summarized by, for example, the output quantity at 24 hours, or it could be the average over the first 24 hours, and then plot that as a function of period. And then you might see one of several possible patterns. And in classical control theory, they have names that are shown here. This is what's called a frequency response, or if it's the period, then I guess you call it the period response. And it, excuse me, it can teach a lot about the system. And so it has been now measured for different areas of biology. And And in a way, I think the talk of this morning fits exactly into this category. And this is something that in, I think, scheduling and dosing of drugs has been particularly fruitful. So this is an unrelated system, but in other systems, they have also found that if you dose in the treatment rather than giving it a continuous signal, then you can get a larger outcome. And this is easiest to respond to understand, I think, qualitatively. Excuse me, if you think about this particular type of frequency response shape where there's a peak, what's called band pass, you see that there can be situations where there's a stronger response if you pause the input. Right. Okay, so this is, people have taught the idea what happens when you do this to a T cell, and so multiple groups have done this. So the idea again is that they vary the on period and the off period, or the overall period, and they measure some output. And what they see as they vary specific T cell type and the specific output being measured, they see basically every qualitative possibility that you could imagine. And this is really intriguing to us, and so we immediately thought, this is quantitative. Can we make a mathematical model that is quantitatively predictive? And the goal that we set out for ourselves is, can we have a model that has a standard residual within the standard error of the mean of the experiments? So can we get within those error bars effectively? Okay. All right. So now I will tell you about the method that we used. And again, I think we're applying this method now to multiple systems. And I think I'm hoping that it is more than just about the specifics that I just showed. And I'll describe it to you in two axes. There's this axis going from left to right, and then there's the axis going down. The axis going towards the right has been worked on by many groups. It's what I would call model complexity, increasing model complexity. And this is, I think, being innovated. We're building on the work of other people. The idea is that we start with a very simple model. So there's an input, I of t, and there's an output, and there are steps in between where we write down very simple ordinary differential equations, ODEs for the time trajectories. In this case, they're all linear. And we have identified the intermediate steps with particular measurable quantities. And then we show that this fails. Okay, and once it fails, then we add complexity by adding more arrows. And then we can do this either procedurally using some algorithm, or we can do it ad hoc as long as the models don't get too complicated, where as we're adding arrows, the equations get more complicated, including nonlinear. And as we add more compartments, if the compartment does not correspond to something measurable, we refer to it as a hidden compartment. And then this, we can keep going in principle forever and make a more and more complicated network. And several groups have worked on how to automate this and rigorize this, including Michael Savageau's group at Davis and Michael Stumpf's group and several others. And so what I will show you is us, for the system that we used, it turned out that we didn't have to go that far to the right on this diagram. And so we were able to still do things more or less by hand. The new thing that I will show you is going down, what I call increasing model flexibility. And this was inspired by a thought of a former student named Ke, who I think shared a thought that many of us share when we start learning about mathematical biology, which is, where do these equations come from? Like this nonlinear equation right here. And that has an answer. Many of these equations are inspired by law of mass action in some kind of situation. Like, for example, if there's an enzyme and the enzyme is saturated, then we get what's called Michaelis-Menten. If there's a multi-step process where you need to proceed sequentially through the steps, this gives an equation that was derived by McKeith in 1995 called kinetic proofreading, and you get an exponent and a nonlinearity there. And all of these equations, I think, have proven over time to be good starting points and good approximations, especially qualitatively. But there's no reason, as we've seen from all of our other research, to think that they are exact. And in fact, some of them don't make any sense, for example, if applied to a two-dimensional surface like a cell membrane. So we started to think, how can we move beyond this, this approximation? Right. Okay, so what we thought to do is we take either individual terms or multiple terms and replace them by a universal function that is subject to constraints. And the constraints, I'll explain why these are important later. The constraints are that it has to have the same sign of derivative with respect to its inputs as the original function. So for example, in this case, it has to be decreasing in W and it has to be increasing in M. And the reason that's important to us is that this will then maintain the same arrow diagram. If we want a cartoon picture of the model, that cartoon has not changed. But what has changed is that now this can select from an arbitrary set of continuous functions subject to this constraint. The way that we implement this is by a universal function that is now well known and well studied. It is the neural net. It's an extremely simple neural net, but it has enough flexibility that it can fit the arbitrary increasing only or decreasing only function. Okay, and then we can procedurally go through and replace all the terms if we want, or just a few of the terms, and that corresponds to moving down. And so we're not changing the arrow diagram, the cartoon of the model as we move down, but we are changing its flexibility. And so this is not this is sort of what it looks like behind the scenes, and there are a lot of codes here that I don't expect you to read, but it gives you a sense of how many models we're actually trying. It is ultimately not that many. It's something that we can do by hand, and as we move to the right, we can move infinitely far. Just keep adding more and more arrows and compartments to the diagram. For example, at this row right here, there are 8 possible models that we could write down, and then as we move down, we're taking the same model, but we're just replacing the terms with universal functions that contain, each one of them contains the ability to span an arbitrary function. And here there's a limit to how far down we can go, because once you've replaced everything on the right-hand side, then that's it. Okay, so I'll show you the result, and of course I wouldn't be here unless it sort of worked. So I'll remind you of the data that is shown in this system. The experimentalists can vary the on period and the off period, and then they're measuring one of the outputs. In this case, it's CD69. What is shown here is the model that we eventually settled on. This was the first model that even qualitatively fit. And it has a name in systems biology. It has a bunch of arrows, not too many. And it has a few enough arrows that I think me and several people in the audience can look at this and sort of digest it. We have names for these types of structure. It's called incoherent feed-forward. And it's able to qualitatively match the data. This is the classical model. It has parameters. It has 6 parameters. And we can search all over parameter space. And what we find is that we can fit any family of off periods, but we cannot fit all of the on periods simultaneously. So qualitatively, we have gained an understanding of the system, but it fails to meet our stringent criteria, which is to actually get within the error bars. And then when KID takes one single term, constrains it to have the exact same signs of derivatives, so the same Jacobian sign as before, and then now we have a model that is able to fit all of the data in terms of being within the standard error of the mean of the experimental measurements, which I'm showing there. Okay, and I think the powerful thing here is that we have not changed the cartoon diagram of the description. Okay, so this is again the complicated view of all of the models that we laid out at the beginning, and what we found is that we do not have to go very far down before we were able to get the fit that we sought, which is to be within the standard error of the measurements. And an important thing is that we tried the model up here, which is the classical version with no universal function, with no neural net, to replace one of the terms, and we would have rejected it. And this makes me wonder, are there cases where we've been rejecting models prematurely simply because we really wanted a symbolic form to be able to write that down in? If we hadn't added this flexibility to the model, we would have rejected it and then moved further towards the right in this diagram. Okay, so one of the things you may be asking is, what does the universal function actually end up looking like after being fit to the data? So this is a slightly different system, but we had the same goal. We wanted to fit multiple conditions, multiple experimental conditions at the same time. And what I'm showing here is the classical approach, by which I mean there's on the order of 12 parameters, between 6 and 12 parameters, and we searched those parameters to see if we could get a fit. The fit is shown here. This is the model prediction for all of the data. There's 440 actually data points, many of them clustered near 0 though, for 4 conditions. And the experimental measurements are up here. And what you can see is that the best fit parameter does not lie perfectly on the line. It has, not only does it off the line, but I think what's more concerning is that it's secular. It's not just random up above and below. There's a concavity here. It's got this what I call chubbiness, and then it's got this kick, this very distinctive kick up at the high output quantities here, what I call a chubby kicker. And if we replace one term, again, this is one term being replaced by universal function, everything else about the model stays the same. then we can fit it much better. This is the universal function. Our collaborator, Omer, has nicknamed these flexi functions, and so we call this flexi1. It's constrained to be increasing only, so it can never go back down, but it has this concavity, and then it has the kick at the end. And when we look at its residual, the difference between the error of prediction is less than the standard error of the mean of the measurements. Okay. Okay, so I'd like to give a sense of why I think that this exercise is valuable. There's something called the variance bias trade-off. I think it's known by other names in different fields, and it's a measure of model degrees of freedom. You can have a model with few degrees of freedom, and that sounds bad because it may not fit very well, and that's true. But as you increase the number of degrees of freedom, you find other problems. One is that there's a high amount of variance, meaning that if you fit to a slightly different situation, either noise or a different biological situation, then you don't get the same thing. This is what's called high variance. This is prone to overfitting. You can correct for that with things like cross-validation. And the real problem to me is that the insights don't generalize. Whereas if you have a very simple model with few degrees of freedom, then it may not be as predictive quantitatively, but you often learn a lesson from that model that translates to another biological system. Okay, so this is well known, and what we have done is not defeated this, because when we add our flexi functions, our universal functions, we're adding parameters, so we're adding degrees of freedom. There is another axis that is subjective, that is totally, I think, individual by individual, which is simple versus complex, hard to understand versus easy to understand. And here, this is, I think, really comes down to what your history is and what you're used to looking at. But many of us, I think, as trained as mathematicians, have the same idea of what counts as simple and what counts as complex. And on this two-dimensional chart of moving to the left, that's adding more arrows to the diagram, and moving down, which is just adding flexibility in the functions, we're adding more parameters either way. And so we're not defeating the variance bias trade-off. We're adding these parameters that allow us to fit the model better, but they have all the downsides, like overfitting, that any other method would have. But because there's this other axis, which is subjective, but it's simple to complex, and it's kind of at an angle, what that means is that we're able to go to a certain simplicity or complexity, a certain number of arrows in our cartoon model. And then by going down, we don't actually increase the complexity of the model. It's still simple in this subjective measure, and yet now we can fit more data. Right. Okay, so just to bring home the point that the degrees of freedom is a rigorous measure, but the simplicity to complex is subjective. This is my conceptually understandable criterion. What is my measure for what counts as simple or complex? And I think it's shared by probably many people here. It's that what I want is some kind of cartoon with arrows, and I really want the arrows to correspond to some kind of enhancement, meaning an increasing effect, or some kind of decreasing effect. And if there's an arrow that has both in it, then to me that's something that is difficult for me to digest in my mind, and I think that's true for many of us. And that means that there's these restrictions on parameters. Other people that I've talked to and shown this to have other opinions about what counts as simple and complex. And so my colleague Omer Dushek, he said, no, actually what we really need is not too many arrows, and the arrows have to not be too steep. And the language that he would use is that they're not switch-like. And this, I think, is understandable if you are a person who studied nonlinearity and try to understand where does nonlinearity come from. If there's too much steepness in the functions, then it feels like cheating, and it feels like you don't actually understand what's going on. So he said, we need universal functions that are constrained by their derivative, and this is often measured in log derivative, and the log derivative has to be less than some critical number. Yet again, there are other people who just want to know what is causally connected to what in the model, and they don't care whether it's an increasing or a decreasing function. And there's a really beautiful work by Gudens and Neuser that collected data and then tested several networks and were able to decipher what the arrow diagram was, even though the arrows might be increasing or decreasing, and they didn't care. They just wanted to understand flow of information. This is yet another legitimate criterion, and I should say that if this were the criterion, then the mathematical tools to study this are much more readily available, and there has been other work in this direction. One of the reasons why we need to build this up from scratch is that we need a catalog of functions that satisfy these particular constraints, and the challenge is they have to be compatible with our learning algorithms. Okay. Okay, so now I will tell you what is the mathematical why is this so difficult, and why I think we've just barely gotten it to work, and why we're still sort of holding back on proceeding to publication. So the challenge is learning, that is finding what is the set of parameters and the set of universal functions that minimize the difference of the data, what you might call the loss. And so that means there's some kind of measure of goodness of fit. In our case, it's the sum square residual, that's the difference between the prediction and the data. And what we are really used to is that this is an extremely rough landscape. And so you can think of this as shown in 2D. Of course, it's more like 6 dimensions or 12 dimensions, not 100 dimensions, but around a dozen dimensions. And it's very bumpy. And finding that minimum requires jumping over all these local minima. There are algorithms that can do this, like simplex-based algorithms, and then there's the Bayesian algorithms like ABC-SMC that we've used and others have used that are really good at escaping local minima. And I used to think that this was it. And in fact, I thought anytime there was a class on like convex optimization, I would skip the class because I thought that this is all we're ever going to deal with. It turns out that that's not true. There are many functions that are very high dimensional, meaning they have hundreds, thousands, millions now parameters, but they have this beautiful property that they're extremely smooth. And it has been proven in many cases that although they may have multiple minima, the minima all attain the global minimum. And that means here, you can just make an initial guess. There's no concern about getting stuck in a local minimum. And then you use a gradient descent method. And two of the popular ones are ADAM and BFGS. And these are quadratically convergent. And so they just work like magic. They take you right to that minimum. The revolution in computing that allows this to work so well is automatic differentiation, the code that can take its own derivative. And what we have is a model with both kinds of terms. And this has been what we've struggled with. So this is a photo of John before I met him. There's John. And these are other mathematicians. This is Sandy, who I've never met, but was the speaker this morning. And so with apologies to Sandy, I believe these pants are what I want to draw your attention to. They're made of a material called corduroy. And corduroy looks like this. It has many deep local minima, okay, that are really deep, which means that they benefit the loss function a lot. But then at the same time, there's other dimensions where, because it's fabric, it might have lots of local minima. And this is now the landscape that we are trying to deal with. It has the curse of high dimensionality, which is fine if all you had was high dimensionality but convexity or degeneracy, like in this case. And then it also has these tons of local minima that you might get trapped in. And this is what we've been struggling with, is this corduroy landscape. Okay, so just to give you a sense of the obvious thing not working. So this is again the same. This is the classical model, and the way to read this is that these are the predictions of the best predictions of the classical model. These are the experimental measurements, and you can see that they don't fall exactly on the line, and they have these secular deviations. This is the one that eventually worked, and I'll show you how we got it to work. It has the chubbiness, the concavity, and then it has the kick at the end. Okay, in the middle, I'll show what happens if we just use a gradient descent method. What you see is that it gets stuck. It goes some of the way, but it's not able to capture the kick at the end, and it's sort of not able to do the chubbiness in the middle. And the reason that it's not able to rearrange the classical points, the classical predictions enough. In fact, if you stare at these dots enough, you see exactly why it's getting stuck. Okay, so all of the algorithms that we've tried, we've found some that work. And I think many of them are like what you would think of if you sat down. They're basically iterative combinations of everything that we've tried. The problem is we don't know when which one will work and when it won't. So one is we've iteratively done gradient descent on the flexi parameters, and then simplex on the classical parameters, and then iterated this. Sometimes that works. We've made a really careful guess to the flexi function, and then done just a massive particle swarm sweep, or differential evolution works as well. And then when we get to a best point, we're still above this dashed line, which indicates the standard error of the mean of the data. That's our goal. And then we do a gradient descent on all the parameters, and sometimes that works too. And so we've been able to make it work in certain situations, but it's super ad hoc and it's super fragile. Okay, so that's sort of where it stands right now. Okay, and then so now I just wanted, this is the last slide, and I'm a bit anxious to show this to you and see what you think. To connect this to some of the work here, on mathematical oncology, many of the models that I've worked on, and by my count, I've counted 7 models so far, 3 from yesterday and 4 from this morning, that fit into a category where there's some dynamics and there are many terms that are modularized. And this is, I think, the power of mathematical modeling. But they fall into kind of 2 categories. Some that are quantitative in their microscopic foundation. These are things like diffusion equations or gradients of free energies. If it's a force balance function, then they're viscous stress or elastic stress. And then there's other terms that are qualitative in their microscopic foundation. And this is things like gene regulation, inhibition, feedback. In many of our models, we have like an active stress term due to myosin, which causes contraction. And here, there are parameters that are meaningful in their direction, meaning that when they increase or decrease, it has a meaning. And so just from some of our work, there are parameters where we know what it means to increase it or decrease it. There are drugs and experimental perturbations that do this, Y compound, blebbistatin. But still, it feels like this is different. And I think now there are many people who are trying to tackle this in the context of making more accurate predictions. So there are several approaches that exist out there. One is forget about solving for you and instead find an approximation that approximates this model and approximates the data. And there's some kind of hyperparameter that this determines how much of which to fit. And this is what I call the PIN approach, because PIN is one of the popular methods to do this. Another method is to don't pick a functional form for F, but rather start with a large library of analytical functions. This is things like trig, polynomials, power laws, and then have some algorithm that picks out a favored one. This is what is called symbolic regression. One of the packages is called Cindy. And what I hope is that our tools can be used as yet a third method, which is you never assume an analytical form. You don't have an algorithm that assumes an algorithmic form. Rather, you just take this universal subject to constraint function, and then you learn it directly from the data. Okay, all right, so that's it. Thanks to all of the collaborators and to our funding, and thank you.
Question?
Yeah.